Okay, so last time we were talking about the different members of the proteobacteria, and then we have a subdivision of these proteobacteria into groups. And then they typically name them, these groups, based on the location of where these bacteria might be, and more so it's about the ribosomal RNA that they share. And then they classify them, and then they put them into different groups, including gamma proteobacteria. And part of the members of these were Proteus vulgaris, for instance, or other types of Proteus, of course. And then we have another group of bacteria within this, or phyla, that it's called Enterobacter. And again, all the gamma proteobacteria, as the rest of the proteobacteria, are gram-negatives. So in Enterobacter, we have two important species, Cloacae and Aerogenes. They produce UTIs and also communal infections. And then we had before, within this Enterobacter group, Chronobacter sakazaki, but they reclassify it based, again, in the presence of specific sequences within this ribosomal RNA. So our ribosomal RNA signature is what it determines whether a bacteria is part of one group or not. So then they reclassify it, and then these bacteria, it is being shown to cause sepsis and meningitis in infants, and it's associated with contaminated infant formula. Now we have a subgroup within the gamma proteobacteria. It's called Pasteurella, and then we have several species like Pasteurella multicoccida. That this one, it can cause in humans infections, subcutaneous infections, after having a bite from a dog or a cat. But this one is more specific to be found in dogs. So oftentimes a person have a bite of a dog, and then of course maybe this dog was vaccinated against rabies, and then the owners the person do not care about searching medical attention. Perhaps they don't wash really well their wound, and then eventually, like within 2 or 3 days, they end up with edematous hand, like if it was that part of the body that the dog bite, and then end up with a lot of redishness and pus. So that's very likely Pasteurella multicoccida. And then we have within these Pasteurellalis, Haemophilus species. And then one important member, it is Haemophilus influenza. This bacteria causes infections, especially in children, that can be life-threatening. Like, for instance, it can cause severe inflammation of the epiglottis, which is like a cartilage that covers the opening of the larynx. And then the child cannot breathe, and he can die because of that. Other types of infections that it can cause is meningitis, which is serious as well, and otitis media. Thankfully, we have vaccination against this, and the abbreviation for this bacteria is HIMV as boy, HIP, Haemophilus influenza type B. Now, this bacteria is called Haemophilus because it requires a specific factor from our blood to live, because it doesn't have certain proteins in the cytochrome type of proteins that we have in the cells, and then it requires factors 10, which is heme, and factor 5, which is NAD and NADP. And Haemophilus stands for philus, which is loving, hemo, which is blood. Now, there is a specific way to provide these factors, 5 and 10. So you put them together in like some discs that they sell with these factors. And then you use as well a type of agar that's called chocolate agar. It's not made with chocolate, it's just like it's semi-hemolyzed blood. And when you hemolyze blood, it looks like a little bit brownish. So it's similar to the blood agar that some microorganism uses. However, this one, it is almost hemolyzed to allow these factors to exit the blood, and then the bacteria can grow in this type of media. And again, thankfully, we have vaccination against this because infections with these Haemophilus implants are very serious. Now, for Delta proteobacteria, and this is the last group that we're going to talk about, but there is another group that is called Zeta proteobacteria. I don't know, they skip the letters in between, and then there is only one member of this bacteria in the Zeta proteobacteria. So we have Delovibrio, which is a bacteria that it is a bacteriovorous bacteria, meaning that it attacks other bacteria. So this is a vibrio, and then attaches into other gram-negative bacteria, enters into the periplasmic space, living there and replicates in there. And then we have the sulfovibrio within the Delta proteobacteria that are part of the bacteria that is helping in the cycle of sulfur. So they can use elemental sulfur or sulfate instead of oxygen for obtaining energy. And then these bacteria, because as any other bacteria that uses sulfur, they are anaerobic. So they live in anaerobic environments, and then they live in an intestinal tract, which for the most part is anaerobic. And something related to this bacteria, no matter if it's the sulfovibrio or not, bacteria that uses sulfur, they can generate hydrogen sulfide or not, but sometimes the sulfur that they use or that they release the other oxidized form of sulfur or reduced, they leave it in a film, and then when there is iron around, you combine sulfur with iron, and then it gives like this dark appearance in the biofilm. So here we have a transmission electron micrograph of Delovibrio. So this is a rod. Here is a gigantic rod, and here is the tiny vibrio with a single polar flagella, and it attaches again into the cell, and then it penetrates in between the two plasma membranes in the periplasmic space and replicates in there. One member of Delta proteobacteria that is used to study the formation of groups in the bacterial world, it is Myxococcalis. The name is coming because it's a coccus, of course, you can see in this ending, and myxo because it forms like a mucus-like film as it's gliding into the surface where they are located. Now, as these Myxococcalis form groups and mature as a group, it forms a group that is called a fruiting body. And within this fruiting body, you have some kind of round capsules that holds inside a lot of spores that we call myxospores. So here is the life cycle of Myxococcalis. So here we have a mature fruiting body. It looks like almost like if it was a palm tree or something like that. So within this capsules that they have on top, they have inside, if you magnify one of these, they have these myxospores. And then when these myxospores are released into the environment, just as any other spore, if they find an area where you have a lot of nutrients, water, things that are favorable for it, they will start forming this vegetative form. So they start protruding these extensions, and then they form these kind of almost like rods. And then these rods, they aggregate and they form a heap. And then the heap grows more as they find more nutrients, and they form like this structure. And this is actually an scanning electron micrograph of how it looks. So it's very organized. And then the cycle will continue. And there's a lot of research done with these micrococcalis to see what it makes them to form these fruiting bodies. Okay, so we're done with the proteobacteria. Now let's talk about other gram-negatives. So we have a group of gram-negatives that are part of the Campylobacter rota, and all of them are rods. They are helical or curved rods, and they are microaerophilic. So they require very little oxygen. So we have within Campylobacter one species that causes foodborne intestinal disease. It's called Campylobacter jejuni. It is causing inflammation in the jejunum, which is the second portion of the intestines. And this bacteria, it is known to contaminate not only produce these infections in humans, but also in cattle. And actually it is found basically sometimes in meat, and then persons who eat this contaminated meat, they don't properly cook, then they will ingest this bacteria, and then they will develop this gastroenteritis. Another species that is not mentioned here, it is Campylobacter fetus. This one causes abortions in cattle and also other domestic animals. Helicobacter. We have in Helicobacter a species that is called Helicobacter pylori that lives in the pylorus, which is the most lower part of the stomach in humans. And this Helicobacter pylori, what it causes initially is gastritis, so it inflames the epithelium of the stomach. And if the infection continues, if it becomes chronic, the person will develop an ulcer. And that ulcer typically become cancerous. So one difference in terms of how does this bacteria look under the microscope is that Campylobacter has only one polar flagellum, while this one, a Helicobacter, has multiple flagella. And now, Helicobacter is this type of infections that if it's diagnosed on time properly, and then you get the treatment, which is sometimes a cocktail of antibiotics, the person will heal right away, and it wouldn't have like greater chances of having stomach cancer just by itself. We're not talking about other factors. Maybe the person had other factors that contributes to that without having an infection with Helicobacter pylori. But something for sure is that there are some tests that indirectly can tell that the person maybe will have this infection, like the breath test, because this bacteria has an enzyme that's called urease. And with this urease converts part of the proteins in the stomach lining into ammonia. So the person might have like a smell or a breath that smells like ammonia. And then they do this breath test, like if they do like the ones for people who can't drive because they were caught with DUI. So that's suggestive of Helicobacter, having a positive test for ammonia in their breath. Definite test is introducing an endoscope in the stomach, take a sample of one of the borders of the ulcer, and then find if you see this bacteria there. I mean, you will do some testing with gram-negative from this sample, and also find if you have malignant cells. And you have to do it in the border of the ulcer, because if you do it in the center, if that ulcer is very deep, that can rupture the stomach, and that's not good, of course. Now, another thing about this is that this bacteria causes cancer because it can inactivate p53, which is a tumor suppressor gene. So it can integrate its genome into the cells of the stomach and then can cause ultimately cancer. It's highly treatable and it's not hard to get rid of. So here we have Helicobacter pylori growing in the stomach lining, which is all this. I don't know why they put it green. I will enhance it with red color. It's a gram-negative. Okay, more about other gram-negative bacteria. We have environmental bacteria that are called cyanobacteria. They are photosynthetic. Wow, yes, very early, sorry. It's photosynthetic, and then it's oxygenic. That's what I was trying to say. So this is the regular photosynthesis process, like you have CO2 combined with water plus light. They give you carbohydrates, hydrogen, and then water. Now, this bacteria has the ability to produce oxygen with photosynthesis, and they may have some structures that they form that are called heterocysts, where they can also help to fix nitrogen. And then some of them, when they live, well, they typically live in water environments, aquatic environments, so they have a gas vesicle that allows them to float into deeper or, well, less deeper and deeper areas of the ocean to capture the light as it's shining into the ocean, and then to perform their metabolism. Typically, they are unicellular, but they can be also colonial, and then the ones that are unicellular, they divide by binary fission. The ones that are colonial by multiple fission, and then they can fragment with their filaments. And then it seems like when Earth in its infancy was developed, these were part of the bacteria that helped to increase the oxygen content and for the rest of the bacteria. So here we have examples of these cyanobacteria. So they grow in groups that we know as heterocysts, and they fix nitrogen. And then here we have a unicellular undergoing binary fission, non-filamentous. So this is filamentous because they form filaments, long polymers of, or not polymers, long groups, linear groups of bacteria. So this is filamentous cyanobacteria, and this is non-filamentous. And then this specific species is called Prokaryotes. This one, it is the most abundant photosynthetic organism. It's very small, but it contributes to produce a lot of oxygen. And this is like the one that apparently helped to create a lot of oxygen for the rest of the bacteria. And now we have other environmental bacteria that is anoxygenic and is photosynthetic. And why is this? Well, because instead of using oxygen as an electron acceptor, they use molecules, like, for instance, hydrogen sulfide. And then they can reduce that into elemental sulfur. So we have two major groups. We have green sulfur and green non-sulfur. Green non-sulfur, it doesn't use sulfur as an electron acceptor. And then this one, green sulfur, uses sulfur as an electron acceptor. Both of them are anoxygenic, both of them are photosynthetic. So we have for green sulfur chlorobly, and then for green non-sulfur chloroflexy. And we also have not only this green sulfur and green non-sulfur, but we have purple sulfur and purple non-sulfur, which are part of the proteobacteria that we talk about not only in this chapter, but also toward the beginning of the semester. And here we have an example of a purple sulfur bacteria forming a group, and these are the individual bacteria. And this is light microscopy. So here we have specific examples of these photosynthetic bacteria. So we have Anabaina, which is cyanobacteria. We have Chloroflexus, which is non-sulfur bacteria, green. Chlorobium, green sulfur. Rhodospirillum, purple non-sulfur, and Chromatium, purple sulfur. And here are which are the acceptors of electrons. When it comes to the non-sulfur, will be organic compounds. It can be including ethanol. Commonality is that they are anoxygenic. Okay, now for chlamydia. Chlamydia is a bacteria that it is kind of unusual because it doesn't have peptidoglycan in the cell wall. And it's a bacteria that lives inside the cell, so it's an obligate intracellular parasite. We have different species. So we have chlamydia and chlamydophila. They infect the cells by the infectious form that it is called the elementary body. And then when the elementary body binds into a receptor of the host cell, the host cell internalizes or endocytoses, and then the elementary body starts forming new bacteria. So we have three important species. Chlamydia trachomatis causes trachoma and urethritis. So trachoma, it is an infection in the eyes. And then it can leave the person blind because it can damage the cornea. Urethritis is an infection of the urethra that we use to eliminate a urine. And this bacteria, as it is growing in the urethra, it can go from distal parts of the urethra into the urinary tract, more proximally in males, and then ultimately will reach the vas deferens and can cause scars inside this vas deferens, which is the tube where the sperm travels. So that can lead to infertility. In females, it does something similar, but that will be in the vagina. And actually, chlamydia trachomatis infection is the #1 cause of STDs in the United States. And then for these infections in females, it doesn't crawl, the bacteria doesn't have anything. We will see how it goes into the reproductive tract. But basically, when it infects the cells, produces a lot of bacteria inside the cells, it will burst, and that bursting is going to seed other cells more proximally, more above the area, and that will make the cells or the bacteria infection to go up. So it goes from the vagina to the uterus, and then into the fallopian tubes. And when it comes to the fallopian tubes, don't think that the bacteria will be in the uterus like, well, I go to the left fallopian tube, I go to the right. No, it will go to both, and then it will cause the scarring of the fallopian tubes and also infertility. So it's one of the major causes of infertility. And it can also cause partial obstruction, and it can cause as well ectopic pregnancy, so pregnancy outside of the uterus. So that's for Chlamydia trachomatis. Now we have this species that is called Chlamydophila psittaci that causes psittacosis, which is a lung infection. And these bacteria live in the intestines of birds. Bird handlers, they run the risk, if they don't take care, wearing gloves, or when they're cleaning the droppings of the birds, to become infected with this bacteria. Now, of course, not all the birds have them. And at least here in the Southwest, we don't have as much of this chlamydia, but northern states, some of the birds that are in the cities, they might have this infection in their, well, they might have this bacteria in their intestinal tract, and then when they release the droppings goes into the sidewalks, and they dry out. And what if there's a wind and someone is talking? Then those particles goes into the mouth of the person, into the respiratory system, and then the person develops the pneumonia. There are other diseases that are happening that way, like Histoplasma capsulatum as well. So, well, that's that histoplasmosis. Histoplasmosis is caused by Histoplasma capsulatum, which is a fungus. So similarly, the birds release their droppings, they go into the sidewalk, they dry out, there's a wind, strong wind, the person inhales these spores or these bacteria and fungus, and then the person develops a pneumonia. And again, thankfully here it doesn't happen because we don't have these birds. However, if you see me walking the sidewalk and I'm doing this, it's not that I'm drunk. I typically don't step on those droppings. I mean, the floor itself is very dirty, so don't step on those. For Chlamydophila pneumonia, causes pneumonia in young adults, in mild form. So here's the life cycle of chlamydias. We have here host cell, this is the nucleus, the cytoplasm, plasma membrane, and then we have elementary bodies, which is the infectious form of these chlamydias. Elementary body is endocytosed, and then that endocytosis create a vacuole. This vacuole will make this elementary body to form a reticulate body, an early reticulate body. And then this reticulate body will divide, producing a lot of these reticulate bodies. And as time progresses, these reticulate bodies become elementary bodies. And when you have several of them, the cells will burst. And that will spread the infection. It takes 48 hours for this cycle to happen. So this is how these elementary bodies, reticulate bodies, and intermediate bodies look in a cell. So here we have the more dense elementary bodies, and then these elementary bodies become smaller, which is called the intermediate body, and ultimately they form this large form, which is the reticulate body. More gram-negatives. So we have budding bacteria of the group Plantomycetes. And these bacteria have a member, which is Gemara obscuroglobus. This bacteria is the exception to the rule of bacteria not having a nuclear membrane. There's always kind of an exception to the rule. Like, for instance, this one. This one has a double membrane around the DNA. So it resembles like a eukaryote, but it's actually a prokaryote. And then mycoplasma. Mycoplasma, well, it doesn't have a cell wall, but it's a bacteria. So again, expect to see that. There are some bacteria like Vibrio that has more than one chromosome. Bacteria typically has one circular chromosome. So here is Gemara obscuriglobus. So here is the cell wall of the bacteria, and this is the nucleoid, and this is the double membrane that surrounds this DNA here. Bacteroidota, we have these bacteria which are anaerobes, and then we have Bacteroides, which is found in the mouth and in the large intestine. So this is very abundant, this bacteria. We have 1 billion of these bacteria per gram of feces. And then we have this bacteria, cytophaga, that it is good for degrading cellulose and chitin in soil. Now, in our mouth, we have an anaerobe that is called Fusobacterium. These bacteria can cause dental abscesses. So here is Fusobacterium. Why it's called Fusobacterium? Because it looks like a spindle. It has taper ends, and it has like this thick part in the middle. It's very abundant in our mouth. And then we have these bacteria that are called spirochetes, that are spiral bacteria. They have the internal axial filaments underneath the sheet. We already saw that before. And when they move like in this rotational movement, like if it was a corkscrew. We have several species that are important. We have Treponema pallidum that causes syphilis. These bacteria, it's also really easily to treat. It responds really well to penicillin, ampicillin, many antibiotics. And one thing for sure is that when someone has a treatment with this, for this bacteria and other bacteria that are gram-negatives, is that as the bacteria is being killed by the antibiotic action, then you will release pieces of LPS, and that causes fever. In the case of the fever that gives to these patients, is called the Harish-Herxheimer reaction, and you have to warn the patient. You will be having these shots of penicillin, but you will have fever, and that's normal. But it's just because these pieces of LPS recess the temperature center in the brain. And then we have Borrelia species that causes relapsing fever and Lyme disease. Leptospira causes leptospirosis. It is found in animal urine. So here is the view of these filaments inside a spirochete. This helps for the bacteria to move in a corkscrew fashion. And then here's the cross-section of this bacteria, showing you that it has more than one axial filament in this case. So those are the gram-negative bacteria. Now let's talk about gram-positives. So the major subdivision of gram-positives is based on the content of Gs and Cs, guanines and cytosines. So we have a group that is called the high Gs and C content, and then the low GC content. So within this high GC content, or firmicutes, We have two species, Streptomyces, that has between 69 to 73% of G's and C's, and then Mycobacterium that has between 62 to 70%. As you can see, then Mycobacterium, it's supposed to be a gram-positive. However, it is gram-variable in reality because, and that's based on the content of G's and C's, because when you stain it, the cell wall of this mycobacteria is so thick that it will prevent the dye to penetrate as easily. And then anything then that is having greater than 60% of G's and C's can be part of this subgroup. And then for the low G's and C's contents, we have Streptococcus that has between 33 to 44% of G's and C's, and Clostridium that has between 21 to 54%. Now for Clostridium, so we have most of these species that are medically important, and then they are endospore producing, obligate anaerobes, and we have Clostridium tetani that causes tetanus, which is going to lead to paralysis of muscles, especially the ones of respiration, and then a person can die out of that. Clostridium botulinum, it also paralyzes the muscles, but in this case, it's not going to be a paralysis due to excess of contraction of the muscles, but all the other way, so the muscles won't be able to contract. So this is called flaccid paralysis. And then we have Clostridium perfringens that causes gas gangrene, which is an infection of the subcutaneous tissue. And then we have Clostridioides, and we have C. difficile, which is a bacteria that causes diarrhea in patients in hospitals, especially the elderly, after they have received a treatment of antibiotics, like, for instance, clindamycin. It's like one of these antibiotics that just because it gets rid of this normal flora, and this is actually, guys, C. difficile is part of the normal intestinal flora. So this one starts outgrowing the rest of the bacteria, and then it causes kind of a membrane in the intestines, large intestines, and the person won't be able to absorb fluid, so that's why they end up with a lot of diarrhea. And then we have this bacteria, Pulopicium. This one is very big, and sometimes it's even larger than some protozoans, so that can be seen with the without the aid of a microscope. Here is an scan electron micrograph of Clostridium botulinum. It produces endospores here, and it produces the endospores, and typically before they release them, put them on one end of the cell. So this is what is called terminal endospore. The other species can be distinguished in the gram staining. Like if you stain some of these Clostridia, you have, of course, a gram-positive rod, but you will see the spores. Even when you stain it with the gram staining technique, you can see the spores. The presence of the spore at different levels tells you what species it is. Sometimes they have it centrally located. This one is called terminal. Some of them will have it between one end and the center, so this will be subterminal. And that's how you know which species of Clostridia it is. And now for Pulopicium, here it is. It's a gigantic bacteria. And if you put it side by side with this Paramecium, which is a protozoan, Paramecium measures a little bit over 100 nanometers, but let's say like in this scale, that will be kind of 100 nanometers, or sorry, micrometers. So it will be 1 2, 3, like 4. So this one measures like around 400 micrometers versus this one. So it's like 3 times more the size of this. So it's very big. For Bacillota bacillis, we have several species of bacillus. These are endospore producing rods. They are typically found in soil. We have Bacillus anthracis that causes anthrax. Anthrax is a zoonotic disease. It can infect cattle and other animals, farm animals, and it can produce infections in humans. So this microorganism, it has been used before as a way of, as a weapon of bioterrorism. There were some mail sent into the Pentagon and to White House during Obama era that contained these endospores of Bacillus anthracis. And these Bacillus anthracis, it is more likely to cause infections when it come in contact with the skin than when it's inhaled or ingested, because we have more receptors in the skin for that. So you require less microorganisms. And of course, thankfully, they always screen this mail as it's coming and they find it, and then they prevent it to spread infections or to cause infections. Bacillus thuringiensis. This one is an insect pathogen. The bacteria has a protein that can kill insects. So this one, it is part of these bioinsecticides that has been developed to prevent damage of crops with insects. And Bacillus cereus can grow in rice and other foods, causing food poisoning. Staphylococcus, they are grape-like clusters. We have several species, Staphylococcus epidermidis, which is part of the normal flora, and then we also have Staph aureus, which is sometimes in some people it's part of the normal flora of their nose and their skin, but for the most of the population, it's a pathogen. It can cause wound infections, and it is often antibiotic resistance. And it produces a toxin that is an enterotoxin or an antigen that is called a super antigen. And it can cause severe inflammation and reactions in the body. So here we have Bacillus thuringiensis. It's a bacillus. It produces an endospore. And then here, this protein, which is called a parasporal body. It's a toxin that can kill insects. So this is like the one that is used for protecting crops. Staph aureus, this is a scanning electromicrograph enhanced with color. It's a coccus, formed clusters, and it causes many infections from superficial to deep. In the skin can cause, for instance, in males that have beard, it can cause folliculitis, because when they are shaving, the blades, they can cause microcuts, and if they have a Staph aureus in their skin, that bacteria is seated under the skin. It can go into these sebaceous glands and the sweat pores and causes inflammation. You can see because they have a bunch of bumps, reddish bumps, and then it hurts for them to touch their skin. So for lactobacillus, another low GC gram-positive bacteria. So these are aerotolerant anaerobes. They are used in the food industry because they produce lactic acid, so they from carbohydrates. They can be used to produce yogurt or cheese, for instance, or any other dairy product. And sometimes they are part of these probiotics. So this can be used, for instance, when someone has antibiotic therapy, and then when someone has antibiotic therapy, something that happens is not only you kill the pathogen, but also the normal flora. So this is one of the ones that can be used to colonize again our intestines. Part of this Lactobacillus species, we also have Streptococcus. So Streptococcus, it's a spherical shape, it's a coccus, and forms chains. Remember, strepto means chains. So we have several species of Streptococcus. We have some that are called beta hemolytic, others alpha hemolytic. Beta hemolytic means like they destroy red blood cells when they are grown in the lab with red blood cells. They destroy them completely. They leave like a white halo around the colony. And then the alpha hemolytic, they partially destroy or oxidize the erythrocytes. So they leave like a light green color. And then we have another type of hemolysis that is called gamma, in which you don't have hemolysis itself. So it leaves like a dark green color when you grow bacteria in blood agar. So when it comes to beta hemolytic, we have two important species, Pyogenes and Agalactiae. Now, before I tell you a little bit about this, so we have within these groups a classification based on Lancefield classification. Dr. Rebecca Lancefield, she discovered several antigens in this Streptococcus, and she gave letters to design these groups. So we have alpha to G groups. So the beta hemolytic are between alpha and G groups. So then out of these beta hemolytic, we have S. pyogenes, which is the cause for sore throat or pharyngitis. And then also this bacteria causes scarlet fever and impetigo. Impetigo, basically, it's like an erythema in the skin, scarlet fever. It gives fever, and also it's an infection that can affect your skin, and it leaves your skin, especially in children, feeling like sandpaper and all reddish. So that's Pyogenes. And then Agalactiae. It causes neonatal sepsis and meningitis. And this is kind of one of the most important species just because it causes as well stillbirth. For alpha hemolytic streptococci, we have S. pneumoniae that causes pneumonia, and then S. mutans that causes dental cavities or caries. And then for this S. pyogenes, or well, the S species in general, Streptococcus, sometimes they are resistant to the immune system of the host because they have a protein that's called M protein. And here is an scan electron micrograph of Streptococcus, so forming chains of coccus. Enterococcus are found in the intestinal tract of the host, and it is part of this bacteria that can infect wounds in patients as well. So it's a nosocomial infection. We have two important species, Enterococcus faecalis and Faecium. They not only infect wounds, but also they can cause UTIs. And they are also feared in the hospitals because they are resistant against many antibiotics. And then Listeria monocytogenes, that causes stillbirth. It can infect the fetus. So is this one of these bacteria that can cross the placenta. And it is found frequently contaminating vegetables. It survives refrigeration, so dairy products or delicatessen can be contaminated with Listeria monocytogenes. So it is not advisable then for ingesting certain foods during pregnancy. Mycoplasma is a bacteria that doesn't have a cell wall, and because it doesn't have a cell wall, it's pleomorphic. Pleo, many, morphos, shapes. So it can take different shapes. And it is capable of free living existence and causes walking pneumonia, which is a mild type of pneumonia. So the person doesn't feel so badly, but still has coughing, and then may have some fever, mild fever. And then the colonies, when you grow them in the lab, they look like fried eggs. That's characteristic of mycoplasma. So here is mycoplasma, pneumonia, growing in tissue. All of this is pleomorphic. It's a group of mycoplasma there. So those were the low GCs, gram-positive. Now let's look at the high GCs. So this bacteria is characterized by being highly pleomorphic, and they can form filaments, and typically they live in soil. Now, I guess here the reference for highly pleomorphic doesn't mean like a single bacteria can have different shapes. No, it means like as a group, the low GCs, the high GCs, they have different shapes, just like the proteobacteria. So basically this is like the proteobacteria of the gram-positives. So the first one is part of the Actinomycota, which is Mycobacterium. Mycobacterium, we have this bacteria, it's kind of a rod, and it has a very thick cell wall that is covered by mycolic acids. These mycolic acids is a very thick waxy material that helps the bacteria to survive the attack of the host immune system. And typically grows slow, but there are some species that can grow faster. And then they require specific nutrients to grow. So they are kind of fastidious in a way. We have two important species. We have Mycobacterium tuberculosis and Mycobacterium leprae. Mycobacterium leprae is not prevalent here in the United States. It's more prevalent in areas of Africa, the Middle East, and they cause leprosy, which is a skin infection that sometimes leads to big pieces of skin falling off from the person and also deform the shape of the face of the person. They take like something that's called like lion-like appearance. Even their eyebrows, sometimes they disappear, and then they have like very thick eyebrows, so that's why they look like, well, not eyebrows, like the skin underneath, they look like they were lions. Mycobacterium tuberculosis. Anytime that we hear tuberculosis, we just think in the lungs. Well, that's one form of tuberculosis. And then, but we can have kidney tuberculosis, bone tuberculosis, and these bacteria, basically, well, all of them, they are obligate aerobes. So, in the case of lung tuberculosis, this bacteria grows in the area of our lungs where we have more oxygen, which is the apex, the top of the lungs. And then with chronic infections, it causes cavities in the lungs. Nocardia. This bacteria lives in soil. typically form filaments. These fragments, sorry. It looks like if it was a hyphae from mold. And it causes nocariosis. These bacteria, we have one species, for instance, Nocardia asteroides, that causes infections in the lungs. And you can also form or get some infections in the skin of the foot and the hands. That is called mycetoma. So that can lead to having like these big toes, or the hands, they become very big, and the fingers, because of the infection with nocardia. Now, Corynebacterium. Corynebacterium diphtheroides or diphtheriae causes diphtheria, which is an infection of the upper respiratory tract that also can lead to death in young children especially. This is part of this group of vaccines that it helps to prevent these infections, together with the one that causes whooping cough and also tetanus that is given to little children. And then we have Propionibacterium, that we have different species, including one that produces propionic acid, but we have other ones that causes a skin infection. And then Cutibacterium causes skin infections. Cutibacterium acnes play a role in development of acne. Gardnerella. Gardnerella vaginalis causes vaginosis or infection in the vagina. It's highly pleomorphic and is gram-variable. This bacteria causes a discharge in the vagina that is whitish in color and is malodorous. Sometimes it can be confused by an infection with Candida albicans, which is a yeast that can cause also vaginosis. Frankia, it's a bacteria found in the environment. It helps to fix nitrogen, so it's good for the plants. Streptomyces, this is another bacteria, together with nocardia, can cause these mycetomas. In this case, it looks very similar, so the person will have the deformation of the toes and the fingers. Not necessarily has to be both, and not necessarily has to be bilateral. It can be unilateral. It can be only in the foot or only in the hands, depending on how the person gets in contact with these bacteria. But we have a species of Streptomyces. It's called Streptomyces israeli, and then that causes also similar infections to nocardia. Now, Streptomyces is one of these bacteria that naturally produces antibiotics. So actually, most of the antibiotics come from Streptomyces. And then Actinomyces forms filaments called hyphae. It is found in the mouth and in the throat. And then here is the view of Streptomyces. So we have these filaments, and these filaments extends these extensions that look like spiral at the end, and these will be filled with these little round structures that we call conidiospores. And these will be the hyphae, or the filaments. It's not hyphae itself because it's not a fungus or a yeast, sorry, a mold, but that's kind of an analog. And then here is Actinomyces. It also forms filaments. Less longer than this, Streptomyces, but it's a filament, and they form groups like this. Environmental bacteria that are high in GCNCs, we have Deinococcus radiodurans. I referred to this before when I was talking about how sometimes we use the knowledge that we gain from studying these bacteria in our benefit. So these bacteria, Deinococcus radiodurans. Well, it's a coccus, and then this bacteria is called radiodurans because it resists radiation. So it has some enzymes that are called recombinases, and with these recombinases, when they are under the attack of radiation and their DNA changes, they are able to excise these pieces that were mutated and put new ones that are non-mutated, so it can repair the mutations. So there are a lot of research done to see if that can help us to swap pieces in our DNA when it's mutated and when we have proneness to have cancer or any other diseases due to these mutations. Thermus or Thermophilus aquaticus. This is a bacteria that was discovered in Yellowstone National Park in the hot springs. This is the source of Taq polymerase, the enzyme that can amplify DNA in PCR, and that's why we have the name Taq, Thermophilus aquaticus polymerase. Now for Archaea. So Archaea, well, we know that they have similar characteristics to bacteria, but it has also a big distinction, not only based on this ribosomal RNA signature that subclassified as this group, but also because it doesn't have peptidoglycan. Instead of peptidoglycan, its wall has pseudomurine. And Archaea lives in the environment. It doesn't cause infections in humans. It's a prokaryote, and in the environment lives in areas where you have extreme of temperatures or excess of salt. So these are classified in general as extremophiles. And based on where they live is how you classify them. So halophiles will be the ones that requires a concentration of salt that is greater than 25%. So that's very high. And then for temperature greater than 80 degrees Celsius, so these will be the thermophiles. And then if they grow at a pH near to 0, these will be acidophiles. And then we have a specific group of these bacteria, these prokaryotes, the Archaea, that produces methane gas. So these will be methanogens. And some of them can be part of the human microbiota in the colon, vagina, and the mouth. But it doesn't produce infections in us. So this is an example of bacteria growing in a surface area. Okay, so lastly, we're going to see some discoveries that illustrate the range of diversity. So we have bacteria that are subclassified as nanobacteria that measure between 0.02 and 0.08 micrometers. And then we have a bacteria that is very large, that's called Thiomargarita magnifica. Its length is about 1 centimeter. So researchers, when they find it out, they were so excited because, I mean, 1 centimeter is this big, right here. So this is one of the exceptions to the rule. And then they also found one bacteria that is called Carsonella rudi, that it has a genome of only 182 genes, which is very small. And also they have found that at least 30,000 species has been described, and out of those, 10,000 or more bacterial species per gram of soil were found in only 1 gram of soil with PCR. And then we have millions of species, of course, to be discovered. That depends so much, of course, on where do we find or where we are seeking for this bacteria. For sure, there has been bacteria found in the Arctic and apparently even outside this Earth, in Mars or so. They have found apparently bacteria. So here we have an example of this bacteria, Thiomargarita namibiensis. This bacteria is very large and it has inside a lot of sulfur granules that when you use light microscopy, they shine. It looks like that. Okay, so that's all for this chapter.